Good morning, ladies and gentlemen. Thank you for gathering here today to listen to my talk. I am going to present you my master thesis project, dedicated to development and application of recombinant system and reporter virus models for Herpesvirus SA8. Now, when we hear the word herpes, we generally think about the cold sores on our lips, which are quite nasty. The cause of these cold sores are human simplex viruses 1 and 2, in short HSVs. Funny thing is that these viruses are not actually interested in our skin, as cold sores is just their way in and out of our body. They take permanent residence in our nervous system. HSVs infect neurons and travel along them to ganglions. No need to worry though, HSVs almost never go further, as there is a barrier between peripheral and central nervous systems. The barrier is what keeps our brains safe. Humans and their herpes viruses have been co-evolving for thousands of years, so we have learned to peacefully coexist. Well, more or less peacefully at least. HSVs, however, are not the only herpes viruses in the universe. There is herpes B virus as well. This one was co-evolving with rhesus macaques, and it lives peacefully with them. Unfortunately, when it comes to humans, the peace treaty is over. It turned out that the B virus can infect human neurons as well, and this is a huge problem because the B virus, unlike HSVs, manages to cross the barrier to central nervous system, reach the brain, and cause severe encephalitis, which leads to death. Luckily, people working with macaques are now aware of the danger and are taking necessary precautions. What makes the situation curious from the scientific point of view is the existence of another monkey herpes virus called SA8. 83% of SA8 genome are identical to that of the B virus, which is a lot to have in common. Surprisingly though, despite the similarity, SA8 cannot be transmitted from monkeys to humans. It simply doesn't infect our cells, which raises a question. If those two are so similar genetically, what makes them so essentially different when it comes to humans? But to answer this question, researchers need some better tools and models, and development of such tools and models is exactly the purpose of my project. The best way to speed up research in virology is to create a new version of the virus which is easier to track. To do so, special genes are inserted in the genome of the virus, for example, genes of fluorescent proteins that make the virus glow. The problem is that the genomes of herpes viruses are untypically huge, and thus this is hard to insert anything in them. In scope of this project, I was testing a novel approach to genome modifications, called a phosmid library. For the reasons of safety, this approach was first tested on the SA8 virus. So, how does this phosmid library approach work exactly? First step is collecting a huge number of SA8 viruses. I isolated their DNAs and cut them into pieces. These pieces I fused with special carriers called the phosmids. This process was repeated many times to get as many different phosmids as possible. As the DNA insertion is completely random, I cannot know which part of the genome is located in which phosmid, so I had to check them one by one in order to select the phosmids of interest. The purpose of this selection was to obtain a set of phosmids that would altogether carry the whole SA8 genome. This goal was successfully achieved. My set consisted of 5 phosmids in total. The benefit is that I could work with pieces of SA8 genome individually and separately, using the phosmids, instead of working with the full SA8 genome altogether. This considerably simplifies the workflow. As a result, I managed to insert some reporter genes into a SA8 genome relatively easy. Good job, me! But what comes next? The next step is the so-called virus rescue. I have put my set of phosmids, including the one with the insert, into living monkey cells. This basically imitates the normal infection by SA8 virus. And just like during normal infection, the cell starts producing new virus particles. However, thanks to the modification in the phosmid, the new viruses obtain some extra properties, for example, an ability to glow. That is how a reporter SA8 virus was generated. Or, to be more specific, reporter SA8 viruses, as I have made several types of them. Such a reporter virus model significantly simplifies the research. For example, it is easier now to detect the onset of infection and its spread, as now the infected cells are glowing under the microscope. In addition, it is now possible to understand how the virus affects individual cells during the infection, as everything is visualized. You just need to take a look. Using such reporter viruses, one could test potential antiviral drugs or study several viruses in parallel in order to understand their differences, which is exactly the plan for SA8 and B virus. So, the take-home message is, the phosmid library approach was proven to be effective for Herpesvirus SA8. I managed to insert reporter genes and get my glowing viruses. If you want to learn more details on how this can be done, I encourage you to read my article. Now, the same procedure can be repeated for other herpes viruses. For us, it means repeating this for the B virus. But that would be the topic of another thesis. I would like to thank my colleagues from Professor Perlman's lab and, above all, my supervisor, Dr. Michael Winkler, as without him this project would never be possible. And also, I would like to thank all of you for your attention. I hope you enjoyed my talk.